Bismillah Rahman Rahim. Welcome to my channel. This is Hafiz Muzammil Rahman, and we are going to discuss a new topic, and that is very important and interesting as well. That is protein structure prediction. If we have a sequence of any unknown protein, so we can predict the structure of that protein by its sequence. How we do this? We simply look at the sequence of the protein and find out the structures which have somewhat similar sequences, like I have like the sequence which we have. I mean, if you have a sequence of any protein that is unknown to you and you want to predict its structure, you will have to find out those structure which have somewhat similar sequence related to your sequence, and that is called identification of the templates. Templates mean if the structure have similar sequences, their structure will also have somewhat similarity between them. So the first step is sequence similarity with the other already available sequences. And then when you will find out the template which have somewhat similar sequence to our sequence, then we will align those sequences of the template to our sequence. And after the alignment, we will have an idea how much these sequences are similar to our query sequence. And then we will select the template which have some good quality, which will have some good features. We will select the template according to our need, and then after the selection of the template, we will go to predict the model of our query sequence. And then after modeling, we will evaluate the model. If there is some problem in the loop, we will go for the loop modeling, we will go for the sidechain modeling, and we will optimize our protein, our predicted model, and then we will evaluate our model whether that model is good for or not, whether we can go further to drug discovery or docking by using this model or not. So I'll perform some experiment, or I'll perform in front of you. If you have a sequence, like if you open a UniProt and you find out any sequence which you have, so I will work on this protein sequence NS7A of the SARS-CoV-2. So this is the, in the end, this is the sequence. I will go for the FASTA format and copy the sequence. So this is the sequence for which I want to model the structure. Suppose we don't have the structure for this specific protein, so we have just the sequence of it. So I will go for the modeling of this sequence. For this, we will use a very famous server that is called HHpred. Just type on the Google HHpred and this page will be opened. The first link will lead you to its home page. So I have clicked on this. So this is the home page. In the home page, you will find out this input. When you will enter over here, you can paste your sequence here. So this is the sequence. You can adjust it or if you don't, no way, no problem at all. So by pasting your sequence over here, you will have to submit it, and you can check the left side. There are two jobs which I have already prepared for you, because sometime it may take some time, so therefore I have done some work earlier before the recording this lecture. So you will have to submit it like this. So it is, we have found identical copy of your job in our database because I have already done somewhat about this sequence. So this is the yellow, this is the job which I have submitted. What you have done, you have just paste your sequence and then submit it, and then this window will open. And after this, when this window will open, you will find this page. In this page, your sequence, according to your sequence, the server showed some template. The server has shown some template, and it is there is the score against these template as well. Like this is the probability. You can check the sequence. This is your sequence, and this is the template which is covering this sequence. So if you click this template, these are the templates. These are the templates. If you will click any one of the template, you will directly go to over there. Let's suppose I click on this template. This is 6Q5U underscore S. If I click this, so this is the structure. This is the template. So if I click the first one, and that is the template of this. This is SARS ORF7A accessory protein domain beta sandwich structure blah 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 blah, and it has a probability of 100%. E value is very good, and this is the alignment, and identity is 88%. It means you have the amino acid which are 88% identical to this template, and this template is good, so I'll select this template. This template has been selected, and then after the selection you can move towards model using selection, this tab. What we have done so far, we have just we have just pasted our sequence, and then after pasting it we have submit over here, and it shows the results, and like this, these are the result of the templates. The server has searched a templates database against your sequence, and then those templates which have somewhat similarity with your sequences, it has shown over here. So then we have chosen the best template which have very good similarity or identity with our sequence. We have choose this, and then we are moving, after selecting it over here, we are moving for the model using selection, this tab. So I have entered this tab, model using selection, and your job is yes. So it have give me this page. Now they have the query sequence and the selected template, they have aligned, and then I will forward this file to the modeler, because HHpred using the modeler. So in your case, when you will use it, here will be a tab, modeler key is restored in your file. Here will be a tab. This is the PIR of the protein which you have selected, the PIR format which you use in the modular. So you can say this is an automated modular. So you are using the modular in an automated way. So therefore this server is a very good server. So you don't have any need to download the modular, and then you will have to work very tough, and the installation, another thing in modular, they are very tough for the beginner. So if you are a beginner in the homology modeling or threading field, you want to model your structure for good quality, so you can use this HHpred. It is a very good server. It has very good citation as well. So in your case, when you will submit your model over here, so here will be a tab, then you will have to enter the modular key. And modular key, to get the modular key, it is very easier. You can register yourself onto the modular by going their page, and they will send you a key for modular in your email when you will register. I'll try to do somewhat for you if it goes good. Modular license key. So there, this is like that. Yes. So you will have to register yourself over here by going to the official page of the modular. So after registration, when you will agree and accept it, they will send you a key. So this is the key. This is the home page of the modular. So you can get the modular key by registering yourself with them, and they will send you an email. So that key you will have to put over here. You will have to type the key or the password over here. That is called modular key. And then because I am already using it, so when you use modular first time, it demand a key, and for the next time it remember your profile. So there is no need to give the key every time. So therefore it is very good. So after doing this, you will submit your your sequences to the modular. So yes, oh my god, it's so fast. So yes, it is loading the structure. Job is done. In a while it will show you the model over here, like this. Yes, this is the model. So this is the model which you have prepared by HHpred by using the modular. So this is a very easy way to predict your structure for a protein sequence in a very fast and easy way. And then this is the model which you have predicted. And the next phase is, now you will have to check or validate whether the quality of your predicted model is up to the mark or not, whether it is good or not, and whether you can use this model for further study or not. And you will have to check this. For checking this, you can download the PDB. If you will click on here, it will download this PDB. So I have already downloaded it. So this is the structure which I have already downloaded. So you will open into the Chimera. So what is Chimera? Chimera is a visualization tool. You can also install Chimera for visualization of your protein. So I'll recommend it is a for you to download it. Chimera tool download. So this is over here. No, this is not the official page of Chimera. You can Chimera, University of California, San Francisco. This is their official page. Yes, this is the official page. Okay, you see SF is the University of California page. So this is their official page, and you will click here into the official page, or alternatively you can click directly over here to download it. So this is the download page of the Chimera according to your system. If you have a Microsoft window, you can click this and you will be able to download it. If you're Mac user, then you can click over here. If you have a Linux system, you can click over here. It's very easy. And if you will click, and then you will find somewhat like this. When you will install it, such icon will appear, and when you will again click on it, then it will be installed. It is very easy to install the Chimera. So after installing the Chimera, and Chimera has a many functions. It is a very good tool. You may have, if you work on bioinformatics or in silico studies, you may have heard about the Chimera or PyMol. So PyMol is a very good tool again, but I love Chimera. So I'll recommend for everyone to do install Chimera for visualization and other studies. So in my next coming lectures, I'll elaborate the Chimera as well, and I'll try to record the lecture what we can do in the Chimera as well. So after installing the Chimera, you can open your structure over there. So yes, this was the model which we have predicted by the HHpred or by homology modeling. So after showing it over here, you can click into the favorite, and then if it is difficult for you, and if you're feeling it is somewhat difficult, so yes, by clicking over here, you can open this with the Chimera. So yes, the Chimera is has opened your model, and it is showing one model, and it has 655 atoms and 82 residues. So it is showing somewhat detail the structure which you have opened it. After this, there is the favorite tab in the interface of this Chimera, and when you will click it, the first option, that is model panel, just click on it. So when you will click this thing will be clicked, already checked. There is different option, activate all, etc, but we are not concerning at this time about these things. So just click to the Raman Chandran plot. So Raman Chandran plot is a plot which guide you about the quality of your model. It has these four, you can say, areas. The first circle, this is the most favored area or region. So this is called most allowed region or most favored region. So out of this is the allowed region, and this is this white region is the not allowed region. So we can see that our most of the amino acids or the residues, they are in the allowed region. If you hover your mouse over these, so they can tell you the detail of the axis as well. How Ramachandran work, or how Ramachandran can assess the quality of the model, we will again, it is a very different topic, so we will record a lecture on Ramachandran as well. But in a simple way, if you have an amino acid, so amino acid make two bonds, the central carbon make two bonds or the alpha carbon with two different group. Like the alpha carbon make one important bond with the amino group and the other important bond with the carboxylic group. The amino group which bonded to the central carbon or alpha carbon, it make the phi bond, and the carboxylic group of the amino acid which make the bond with the alpha carbon, it may make the psi bond with it. So you can check whether these bonds are corrected, what is their angle, and how many residues are in the favored region and how many are in the allowed region or how many are in the disallowed region. If your protein has 90% or above, you the if you predict your model and you check the quality of the model by the Raman plot and it shows more than 90% residue in the most favored region or allowed region, so it means your protein quality is good. So we will record a lecture separately on the Raman plot as well. So this Raman plot shows our protein is good. So we can use this protein for further studies as well. So there was online server as well that was called ProCheck. Let me check whether it is working or not. ProCheck was a server which yes. So you can do like this. You can upload your model to this ProCheck web server. It is a very good web server which identify or which validate your protein structure as well. So I'm just trying. I have just clicked. It has a different feature like array, verified 3D. And there may be lot of analysis as well. So we are interested in the ProCheck analysis. We are going for the results. Yes. Where are the Ramachandran plot? Yes. This is the Ramachandran plot. So these red region, they contain the most favored. This is the most favored region. If the residue are present in this region, it means your residue are present in the most favored region and the quality of the protein is very good. As you can see over here, that is our 69 residues are present in the most favored region, ABL. This is A, this is B, and this is L. This is capital A, the red in the red region. This is capital A, this is the region capital B, and this is the region capital L. So these region, they are the most favored region, ABL, and they contain 69 residues out of 82. Out of 82 residue, 69 are present in the most favored region. So it means this contain 97.2% residues in the most favored region. So it means it's a very good region. It is a very good model. And only 2 residues, they are in the allowed region. 2 residue, they are in the allowed region, like this. A, B, L or I. This is small a, small b, this is P, and this is L or I don't know. Okay, so and then if this yellow region is the allowed region and this somewhat light yellow, this is generously allowed, and and the other, if your residue are present in this white region, it means they are in the disallowed region. So by this Ramachandran plot, it shows our structure is very good. So you can download this picture for your publication or your thesis over here. So you can click the PDF file, so it will be downloaded. So this was the homology modeling of the protein by HHpred. So now we can use another tool, that is the SwissModel, for the same homology modeling. If we have a model, we have a sequence of protein, this, we copy it and we go to the Google and type SwissModel. So yes, we have typed SwissModel. So you can click, this will be the page of the Swiss modeling, and then you can click over the start modeling. Yes, so when you will click on the start modeling, so this page will appear. So you will have to paste your sequence over here. So we have pasted our sequence over here, and then before going to the building of the model, you will have to, what was the steps? I have told you the first step is selection of the template, identification and selection of the template, and the next step will be, you will see how much your sequence is aligned to the selected or identified templates, and then you will decide which template you will use to model your protein. So I will click on the search template. So it is searching for the templates. I have already done some work on it. Okay, so yes, these templates will appear when you will search. When you will search template, there will be different templates which will appear here, like this. There are many templates which have appeared. 15 templates have appeared against your sequence. So in these templates, you will have to select the template which have some good feature and qualities. The first feature is GMQE, that is Global Model Quality Estimate. The value of GMQE is between 0 to 1. Closer the value to 1 is better the model. And the next is, I'll tell you later about the QSQE. And then next is identity. Identity means how many amino acid is identical present in the template to your sequence. So this is the identity 100%, and here is the identity again 100%. So I have choose these two. You can choose 2, 3 template, 4 template, 5 template according to your need. If you think your structure, your protein is somewhat related to these structures, so you can choose these. I have chosen these 2 structures. You can choose more, no problem. So after choosing these 2 structures, when you will click on these, you have selected 2. So these 2, when you will select these 2 template, so here 2 will be shown, and you will click to build the model. When you will click to build the model, I think, yes, so here Yes, it has completed. So yes, if you choose, like I have chosen this one 100%. Only identity is not the criteria to select the template. This GMQE is also important. Identity is important. Similarity is important. There are other factors as well. So I have selected these two template. You can select this one as well. It has a QME is very good. So 3 template has been selected. So I will click this to build model. So this will starting to build the model, and it will appear like this. Yes. So these 3 model have predicted your protein against your sequence. So by clicking this picture, the model will be shown over here. So I have clicked on this. All of 3 has been selected, and you can see over here the 3 models are superimposed with each other. So you can check the quality or assessment of the model as well, and you can check. This is the okay, already selected. I'm just going to change this. Yes. By clicking over here, you can select the cluster. So it will color the residues accordingly. So when you will hover your mouse over here, so it will show you the residue accordingly. Like these residue have different color, and it has this is the alignment. Your query model, this is your model which has been built by the SwissModel, and the second one is the template. This is the template which you have selected to build or to predict your model. So when you will hover over your mouse over here, so they will show the residue as well, and if you click, they will show the residues accordingly. So in this way, you can predict your model. And then the next thing is, you can assess the quality of the model as well. The quality of the model can be assessed through these 3 things. This is Q mean Z score. This is important. If the value, if the quality of the model is good, above this is the threshold somewhat. If it is above 6.6, the quality is good. Or some people say it is above 7, the quality is good. So from here you can check the which residues are below the threshold as well. Like they have selected the threshold 0.8, so you can check the quality or the residue, like the last residues 88, 82 and 81, they have they are below the average. And these are the Q mean, the quantity, the quaternary structure interaction. It shows the beta interaction. It shows the all atom interaction with the other. It shows the solvation. This shows the torsion angle. So collectively, all of these scores are shown in this plot. So this is called Z score. It has 3 areas. First one is the dark gray, and the second one is the light gray, and then the white area. If your protein is good, it will be lie into this dark area. if it is present in the dark area, your model is good. If it is present in the light gray, it is somewhat good. But if it is present outside these two areas, it means the quality of the protein is not good. Again, you can also assess the quality of the models. This is the one model, this is the other model as well. You can select the model by checking all these criteria, and then you will decide which model you are going to use for further analysis, for further study, for docking or somewhat else. So you can click here the structure assessment by checking the Raman plot as well. You can directly check the Raman plot in the Swiss model. So this is your model and this is the Raman plot. And just look, if you will hover your mouse over here, so these are the residues which are present in the mostly allowed area. So this is the psi angle, this is the phi angle, and these residues are present in the most allowed area. So you can check. The Raman favored is 96.88%. It means you can use this model for further analysis as well, and you have these assessment over here as well. So overall the quality of the model is very good. So these two server, they are very good for the modeling of your protein by homology modeling. And two other server, they are very easy, and I'll just conclude it into the next 5 minutes as well. They in these two which we have used, that is the HHpred and the SwissModel, we'll have to search the template by ourselves, and then we have to select it. We have to select the template by ourselves as well. But if you think it is a laborious work and you are not sure which template will be good for your protein, so you can use another server that is called RaptorX. So you can go to the Google. Where is Mr. Google? Yes, you can go to the Google and you can type RaptorX. RaptorX is a very good server for protein structure prediction. It has many different options. You can go and you can download their history. You can download not download. You can just click over here to about, and then it will tell you how this server work and how they have prepared this server, and so on. So you can read about RaptorX as well from there. From here, if you click on about, they have some paper as well, and you can read these papers about this server. So it can predict the structure, it can predict the property, it can predict the contact, and it can predict the structure alignment as well. So so now we are interested into the structure prediction. So you will click on this. There is no need to worry. You will have to just put your sequence over here. You will have to give the name of your job, like my name is Muzammil. I'm just writing my name. If you have a sequence, you don't want to submit it to some server and you want to you have some privacy, so don't write the correct name of the protein or sequence. Just you can write any name. I have named it like Muzammil. My spell is like MUZZAMMEL. Okay, so then you will have to give your email address, and this is optional. If you don't want to give your email address, you will have to submit it to the server, you can wait on this page as well. This page will be refreshing after some time, and you will get your result on the same page. If you don't have some time and you want to get the email, so you can put your whatsoever your email is over here, and then you can check this. And I'm not providing this time. So if you submit your job over here, so this your job is submitted. So you can check the status of your job. It is still pending, and you will have to periodically check this page after to know the status of your job. And if in either way, you can click, you can save this URL address. For by clicking this, you will be able to get the results. So you will get an email. In email, they will give you a link, and after clicking this, such page will open. So this is the model which I have predicted by the RaptorX, and this is showing there is only one domain. If you have a multi-domain structure, so the RaptorX will show you your protein has multi-domains, and this is the sequence alignment. If you are interested into the sequence alignment, you can check this box, and if you are not interested, you can kept it unchecked as well. So I had checked, so therefore it is showing some alignment over here. So this is the model, and only by clicking here I can save it, this model, and then I'll have to check the quality of the model by the same method. You can go to the pro check or the other server, or you can directly go to the Ramachandran plot and submit your protein over there to check the quality of your protein, predicted protein. The last one is I-TASSER. This is the protein structure and function prediction server, and it is iterative threading assembly refinement. And this server is very good, and it is used for the it is the hierarchical approach to protein structure prediction and protein structure with function annotation as well. So it has many different thing. If you want to do over here, you can explore it by yourself by going to the publication and some other things, but now we are interested to build the model over here. So we will have to just paste the structure, and then this is mandatory to fill. You will have to give your email over here. This email is important, and again, if you have an official email of the educational institute, then you will be able to register. Otherwise you won't be able to register over this I-TASSER. So you will have to provide your educational email over here, and then there are some additional options. And if you let's suppose if you want to give some restraints, so you can click over here to give some additional restraints. It means if you want to if you know your structure or your template, so you can give the PDB ID of that. And if you don't have any, if you want to some restraint, so you can put over here. This is the some other optional options. Exclude some template. If you want to exclude some templates, so you can give their details over here. And the third one is, if you want to specify secondary structure for some residue, so you can choose the specify secondary structures, or you can upload the file. So usually the people do not use these things. After putting your sequence or pasting your sequence over here and giving your email and typing your email over here, you will have to just run this. Click over this run I-TASSER, and accordingly you will get your email within a day or two of your protein. So you can again get the protein from this I-TASSER, and again the same things, you will have to check the structure of your protein by the Ramachandra plot or any such things, like you can use these UCLA DOB lab. So it has many other programs. I have choose the ProCheck, and after clicking this you can submit, or you can upload your structure over here, and then you can run it and you can get the Ramachandra plot. So this was all about the homology modeling from my side. Homology modeling is the way to predict the model of an unknown protein if you have a sequence of that protein. So if you want to learn something more about the in silico studies, the drug designing, the pharmacophore modeling, or any other thing, you can write me an email. I'll give it into the description, or you can just comment over in the description, or by getting the email, or you can comment in the comment section. So I will accordingly record more videos, and you are welcome for your suggestion as well. And take care. Bye bye.